Okay. Moving on. All right, these are one of these charts that we all like, and we'll make sense of it now, so that when you start to study it, you'll say, oh yeah, no problem. B cells, T cells, cytomaturations, bone marrow, and thymus, but the stem cells come from where?
Bone marrow.
Where are the stem cells? Bone marrow, yeah. And then they go to where they're going to be mature. B cells don't have to go anywhere. Okay. Surface markers. We said that on the surface of B cells, the receptors are immunoglobulins. And on the surface of T cells, they're MHC1 and MHC2 receptors. We said that for the most part, if you had to make just a snap decision about whether you wanted to associate B cells more with MHC1s or MHC2s, what would you choose?
MHC2.
Yes, and the opposite for T cells. In the circulation, there are very few circulating B cells. That's why we don't have B cells in our CDC and DIP. There are just not very many of them typically circulating. High numbers of T cells. Those are what most of the lymphocytes in our CDC and DIP are of the T cell line. The receptors we said are on B cells are immunoglobulins, but we haven't said yet which type, which class, which isotype of antibodies are on the surface. Mostly it's IgD. Most of the receptors on the surface of standing up on the surface of B cells are of the IgD class, but some are IgM. The important thing to know about the IgM that serve as receptors on the surface of B cells is that the IgM is monomeric. The circulating IgM has how many parts? It's a pentamer. IgM has 5 pieces, but the surface receptors would be just one little piece of that whole. It's not a full IgM molecule. It's a monomer from the pentamer that's the full IgM immunoglobulin. So if I ask you, what is the major type of antibody that serves as receptors on the surface of B cells, you would say? You would say? Major one.
IgM.
Huh?
IgM.
D. IgD is most of it is IgD. But IgM can act as a receptor on the surface of B cells, but when it is present, it's in the monomeric form. So those are the special things you have to know about that one. We know that the surface receptors on T cells are not antibodies, and they call them TCRs, T cell receptors, not too fancy. Where we said that after they're matured, they go to different lymphoid organs and lymphoid tissue. Where do the B cells hang out in those tissues? Mostly in the outside, the cortex. And the T cells mostly are interior to that, or paracortical sites. Do B cells require antigen to be presented with MHC? No. The antigen can just hook up to the antibodies on the surface, so they don't require APCs to bring the antigen to it. Okay, the circulating antigen can hook up to the antibody on the surface of the B cells. The T cells, no, they have to wait for the APCs to come comes and says, here I am, I've got it out here sticking on the MHC molecule, then it binds and starts to do its thing. What happens once the B cells are stimulated? They mature into plasma cells, and the plasma cells then make the, Plasma cells then make the specific antibody. Yes, you know that. Yes, okay, so B cells, once they are stimulated by its specific antigen, and then the T helper cells start to do their thing, they mature into plasma cells, they grow into this huge clone, and memory cells are formed. Okay, so once the T cell side has been stimulated by antigen, and how do they, what's required for them to be stimulated by antigen? Just said. Yes, it requires that the APC show it to them. They're stimulated, they become then sensitized T cells. There's more of those are formed. They start squeezing out the interleukin, and a memory cell line of T cells also formed. So we usually think about memory cells on the B cell side of the immune system. But remember that T cells also have memory. General functions of the B cell side is to make antibodies. The function mostly on the T cell side is helping. Their helper function causes them to synthesize cytokines. They have killing function, those cytotoxic T cells. They can recognize antigens on MHC1s and say, no, I know this was a self cell, but now they're showing that there's something inside them, and they're exhibiting foreign antigens on the surface, so we can go ahead and kill them. The T cell side also have regulating function. They can down-regulate the immune system because sometimes it's the T cell side of the immune system that can say, okay, all right already. We don't need to make more and more and more of these clones. We've got enough out there. Hypersensitivity is when the immune system doesn't listen. Hypersensitivity is something wrong. We don't like hypersensitivity to happen because too much cytokines and too many interleukins and too much mast cell degranulation is going on. So the T cell line has a line of cells that can down-regulate. Say, settle yourself down. We're okay, we don't need any more. Okay. Antigen is that substance that provokes the immune system, that wakes it up and says, hello, there's something in here that's non-self, and you need to wake up and get in this game. Antigenicity is the property of behaving like an antigen. What determines the antigenicity of an invader or an antigen? How big it is. The bigger it is, the bigger the molecule, the more antigenic. Or the easier the way I think of antigenicity is how easy is it for the immune system to recognize it. Accessibility means, can the immune cells get to it? We already learned that there are certain areas in the body that cells can get to, like the brain and inside the eyeball and things like that, inside testicles, and so there are certain areas that are protected, just kind of sealed off, and it's hard for the immune cells to get to them. So how accessible is the antigen to the immune system. The shape, the more convoluted, the more complex the shape, the more antigenic it is, and how foreign it is. What's that talk? What do you think foreignness means when we're talking about immunity? Foreignness.
Non-self.
Non-self like what?
Like, I'm recognizing you as not being part of me, so you're foreign.
Yes.
Non-self, recognition of non-self.
Okay, yes, that's true. But at least we're the same species, so we're not that foreign. But if you try to receive a pig kidney, that's really foreign, and it's really going to be very noticeable to your immune system. So yes, I'm different from you, but there are things that are really different from both of us, and that's going to be even more foreign and even more antigenic. Good. Okay. So what are the characteristics of an antigen? Well, we kind of talked about it already. It's something that's not us. It's not self. It's something that's somehow foreign. It could be barely slightly foreign, or it can be really, really, really foreign. Okay? The bigger they are, the more antigenic, or the easier it is for our immune system to see them. And there's this size, a molecular weight of over 10,000 becomes hard for the immune system to ignore. Lower than that, you might be able to sort of skate by and not really generate a good immune response. So, if you're trying to devise, create a vaccine against a dangerous human disease, okay, you want to make sure that you don't create a vaccine that has components with a molecular weight of 5,000. Because then you're going to inject it, they're going to hurt, they're going to scream, and they're going to cry, and the immune system is not going to be able to see it. So you're not going to have a really good antibody response to it. So if you make it 20,000 molecular weight, your host, the person you're going to inject this thing in, is going to have a better chance of generating immunity against it. So that's kind of what I want you to know about that. An antigenic determinant is called an epitope. An epitope is a small molecular group that's able to be recognized by antibodies. So that means, notice that the last one, an antigen has many antigenic determinants. So if I'm a bacterium and I'm going to infect you as a host, your immune system is going to notice this epitope and that epitope and this epitope and that. I have many epitopes on my surface. Some are going to be more antigenic than others, okay? Because some are going to be bigger, some are going to be more foreign. But as an antigen, I have many antigenic determinants on my surface. So, if you think about it, you can generate several different antibodies against me, because some of your B cells were born to recognize this epitope, and other ones this epitope, and the others this epitope. Okay? So, although we simplistically think about one antigen, one organism, one antigen, one antibody, that's really not the way it happens. It's really much more complex than that. And as remember we said our antigen presenting cells takes it in and it breaks it up and beats it up in all different pieces. It's going to be on one MHC, it might put this epitope, and it might put this epitope. So the immune system I think is brilliant, because lots of different, you're going at this invader in many different ways. Yeah.
The epitope doesn't have to have a 10,000 molecular weight thing. It's the combination of all the epitopes that weigh more than 10,000?
Well, That's a great question. The antigen, my molecular weight is just ridiculous, huge, okay? And as I get broken down into bits inside the antigen presenting cells to be presented to the immune system, if I present too little, my epitope that I present is too small, the immune system is going to ignore it, okay? So the epitopes are smaller than the antigen, but the epitopes are going to be more antigenic depending on how big it is, okay? Are we okay? Say it again, you okay, we good? Okay, so that's me. I'm a giant bacterium with different epitopes on my surface. And this one is presented on the cell, and the B cell that makes antibody A responded to that one, and so the antibody A is only going to lock down on that particular antigen. But antibody B is also being made, and antibody C is also being made, and epitope triangle may be more antigenic than antibody B, and epitope C may just be too little for the immune system to make any antibody towards. So that's the difference between an antigen giant big thing and an epitope. Okay. Again, we talked about sizes, shapes, molecular weights. Notice that this right here is really big, it's really long, but it's very simple. Like a carbohydrate can be, chains and chains and chains and chains and chains of carbons. But the simplicity of a carbohydrate as an antigen means that that it's not very antigenic. And coming up. When antigens are very, very, very simple, like a carbohydrate, the immune system will make antibody against it. But there's a problem with very, very simple antigens or very simple epitopes, is that they don't generate good memory. Okay, so the antigenicity may also determine how much memory, or if there will be memory to the after the immune system has done its thing. If you have real whole cells, whole viruses and whole protozoans, and they're extremely, extremely antigenic. Foreign human cells, just like we already mentioned, my cells are going to recognize your cells as being bad, but my cells are going to recognize pig cells as being horrible. Plants are going to be recognize this as very different as well. Let's see what we got here. See if it'll work today.
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins and polysaccharides. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants or epitopes. Many different antibodies can be made against a single antigen, each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes. Each different antibody binds only to the correct epitope.
Just a prettier way of. Okay. Haptens are very interesting. Haptens are so tiny that even if it comes from a non-self entity, the immune system will respond against a foreign pathogen is if the pathogen is bound to something bigger. It has to be stuck onto a carrier, and usually the carrier is a protein. Once the pathogen is attached to the carrier, the immune system can see it and it can make antibodies against it. Once the pathogen is attached, the immune system can then respond by making an antibody against the pathogen, against the carrier, or against the pathogen carrier molecules. So it can respond in multiple ways. Oops. This just shows that if you inject the hapten by itself into an immune competent mouse, you get no antibody out. If you bind it to something, then inject it, you'll get response to the hapten. Okay, vocabulary lessons. These are easy ones. Alloantigens are antigens that occur between members of the same species, but not in other species. So if you gave me some of your tissues, some of our antigens will be the same because we are both humans, but they will still be antigenic because of all the surface markers that are dissimilar. Super antigens are antigens that promote an out of control, exaggerated immune response. Exposure to a super antigen can kill you. Now you think we just said we don't want tiny antigens. We want them to be the bigger the better because our immune system will see them. But some antigens create such an outpouring of cytokines, bradykinin, and all those things that we talked about that it's overwhelming to the host. Remember the vasodilation that we said that's caused by the secretion of the degranulation and the secretion of cytokines and all those things. Suppose we're exposed to a super antigen and we just get this huge vasodilatory response. What happens to us? What's that called? What's the horrible hypotension called?
Shock.
And what comes right on the heels of shock? Yes. So super antigens can be really bad things. And we'll talk about some infections that act as super antigens, and you'll understand then why being infected with these particular things is so dangerous. An allergen is an antigen, but one that evokes an allergic response. All the pollens and the hay fever and all that kind of stuff are considered allergens. It can be skin allergens, but they evoked an aggravating hyper sensitivity response. Doesn't really do anything good for us. We think about the immune system as a response at the cellular level that does something good for the host. Allergens really don't. Autoantigens, molecules on self-tissues for which tolerance is inadequate. Well, that sounds really fancy. Say that in other words. Somebody simplify that for me. You're the one who's my talker, so what do you think that might mean? I'll volunteer you.
It's when you attack your own tissues, autoimmune diseases.
Right. We said we want to be tolerant to self-antigen. If we lose tolerance or our tolerance becomes inadequate, we no longer are able to adequately recognize our own antigens as self. So we we allow the immune system to attack it as if it's non-self, as if it's foreign, as if it's an enemy or an invader. This you already know, just put it in giant bullet points. We know that the immune system is a result of an interaction between antigens, non-self antigens, and the rest of the immune response. Okay? Mostly we're on the specific side, so we're talking about white cells that are B cells and T cells. Lymph nodes and spleen concentrate antigens and circulate them so they will come into contact with lymphocytes. Why is it a good idea to concentrate antigens? Why should the lymph nodes and spleen not just be a place for the T cells and B cells to hang out? What might be a good reason to concentrate the antigen? You can't speak this time, so somebody else, yes. Oh.
You're talking about like keeping them like more localized instead of letting them just go throughout the body like food loss and all that.
No, but I want to expound on something you said there. Somebody else give me another, say some words. Why do you think the spleen and lymph node might have as a part of their job concentrating antigen? Oh, yes.
Well, I mean, if you're not circulating throughout the body, you're less likely to have an autoimmune instant, right?
What do you mean?
If you have the antigen circulating your entire body, you have higher chances of actually having anything that you want to do.
All right, think about this. Let's say we stepped on that, whatever it was we stepped on, that was contaminated, and we broke through the armor of our skin, and now the reticuloendothelial system picked up some of that antigen and starts bringing it towards the heart, but it passes through some lymph nodes and passes through the spleen on the way. If it's only a little bit of antigen, the lymph nodes and the spleen can put it through this little multiplier thing. Put it in this little machine, and more of the same antigen comes out. And then it puts out in the circulation to expose the immune system to more of the antigen, without the bug having to grow and multiply and do all those things. It can take a little bit of antigen, expand it, so that the antigen comes into contact with the specific B cells and T cells that were born to respond to it. So what it does is it increases contact with the lymphocytes that can help to get rid of it. So that's one of the upsides. You can take a little bit of antigen that just barely came through the particular endothelial cells, and while it's in there, it'll make more of that antigen, and then get it out to places where immune cells might be, to other lymph nodes. And so instead of trying to keep it localized, you actually want to get it out there, so you can get back, because the infection didn't occur in the lymph node, but that's where the immune cells are that can react and then get the good stuff, the antibodies, back down to the spot where where the injury was. So we don't wanna keep it in the lymph node. We wanna make more of it, expose it to lymphocytes that can really help the situation and get it back out there so it can go and do its work. Okay. Now, you know about APCs. T-cell dependent antigen. T-cell dependent, we haven't talked about T-cell dependent or independent antigen, so we must be about to learn something new. Okay. Processed by phagocytes, processed by dendritic cells, and processing means it beats it up, chews it up, breaks it down into smaller pieces so it can be presented on the outside of the cell by a what? MHC what? Class 2 antigens on the surface. Okay. So the APCs modify the antigen, break it up into little bits, and moves it to the surface, bound to MHC receptors. You just told me it was 2. Okay. Now that it's showing the antigens on the MHC2 on the surface, T helper cells that are specific for that antigen can come by and hook up, and once it does, it starts squirting out these interleukins. Okay? The APC by itself can't do it, but the helper cell comes and it starts to squirt out interleukin 1, and it causes the activation of more helper cells. Interleukin 2, but interleukin 1 says it's secreted by APCs, but one's helper cells can too. Interleukin 2 is produced by the helper cells to activate other B cells. Doesn't really activate. What it does is the B cells that are specific to that antigen, because of this interleukin, they can start to mature into B cells, mature into plasma cells so that they can make, woohoo, we're on it now. And interleukin 2 also causes the production of other T helper cells. So we're going to have, by these things being squirted out, we're going to have more B cell lines, more B cell clones, more T cell clones. We're going to get B memory cells, and we're going to get T memory cells, and we're going to get plasma cells. So all that stuff is going to happen once we get the APC to chew up antigen, to present it on the surface. T cells activated, T cells bind, and then all these other things start to happen. Let's see what this one is.
Antigens are processed differently, depending on whether they originate within or outside the cell. Proteins produced within the cell, such as viruses or self-proteins, are broken down into fragments. Fragments of foreign proteins are antigens. The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class 1 MHC molecules. The combination is then transported to the Golgi apparatus and from there to the plasma membrane. Foreign antigens presented on class 1 MHC molecules stimulate cell destruction by activated T cells. Self-antigens are not recognized by the T.
If we're talking about MHC class 1 molecules, what must this cell be? MHC 1 molecules are on the surface of what? Surface of all, come on, all nucleated cells, so we know they're not on. All right, so now if we have a virus inside this nucleated cell, it was a self cell that was perfectly fine and the immune system was happy to ignore it. But now there's a virus or something inside it. It's been broken down. It's gone to the endoplasmic, the rough ER, and brought over to the Golgi, and then hooks up to the MHC class. What did they say? 1. Okay, and so yeah, we know that that was a normal cell before it got infected with a virus. But once it was, the antigen was processed and then hooked up to an MHC1 and presented to the outside. And it looks like we got the T cell now to notice it.
Cells and do not stimulate cell destruction.
Let's back up.
A different process occurs if the antigen originates out as one MHC molecules stimulate cell destruction by activated T cells. Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the antigen originates outside the cell. Phagocytes, such as macrophages, ingest foreign particles such as viruses and bacteria by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class II MHCs, and the two structures combine. The MHC class II antigen complex is transported to the plasma membrane. The displayed MHC class II antigen complex can stimulate other immune system cells to respond to the antigen.
When it said, well, let me see if I can just get it back.
Such as viruses and bacteria, but
How did that get inside that cell? Huh? Phagocytosis, and what helps that process to happen? We started the class talking about that. Anybody, just say something. It's okay to be wrong, but everybody's gonna be right by the time we finish. What enhances phagocytosis? Complement is one of the things that can enhance phagocytosis. And which arm of complement does it? Come on, keep it going. Say it. Look at the cheat sheet down there.
C3b.
C3b, yay! Okay, there's another thing that can enhance phagocytosis, and sometimes on the surface of phagocytes or B cells, there's these things standing up on it. And what are those things?
Receptors.
Huh?
Receptors?
What kind of receptors? That look like this.
Antibodies.
Yeah, antibodies can help the oxidization process too. And we said it's the FC portion that stands up that can hold on to it so that the phagocytosis can happen. So there's some things that are happening beneath the surface that you already know. I just wanted to keep bringing it back to your
by endocytosis.
to your mind.
The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class II MHCs, and the two structures combine. The MHC class II antigen complex is transported to the plasma membrane. The displayed MHC class II antigen complex can stimulate other immune system cells to respond to the antigen.
Okay, now the T helper cells can bind them and all that stuff. Okay, there's that T independent and T dependent language again. Now, I talked about very briefly that some antigens like sugars, like carbohydrates, aren't very good antigens. They are T-independent antigens. And what did I say about those kinds of antigens? What was special about those kinds of antigens? that they don't generate what? Memory. They don't generate memory. Why don't they generate memory? Those kind of sugary kind of antigens? Because they don't have, the T cells aren't involved, okay? They can make antibody without involving T helper cells. And when T helper cells are not involved, those interleukins aren't formed, so they don't stimulate clones and they don't stimulate the formation of memory. So T-independent antigens are not very antigenic. They can get through their current predicament, but they don't go through all this process that involves T helper cells, so there's no memory involved. Okay? T-dependent antigens are more antigenic. The B cell this time is the antigen presenting cell. And we know that on the surface of B cells, antibodies serve as receptors. Which classes of antibodies serve as receptors on B cells? IG and M little. Right, the monomeric M are the kinds of antibodies that are standing up there holding on to antigen green thing. Okay? And once it's brought inside the cell, by the phagocytosis thing that we were talking about. It's processed, it goes through the Golgi apparatus and all those things, and it's presented on the outside of the APC on an MHC2 class molecule, and the T helper cell then can see it, and it starts to release lymphokines, interleukins. And when it does, it causes a clone of memory cells that can respond to green thing antigen. These will mature into plasma cells and make specific IgG against green thing. And it also will make a clone of memory cells, a clone H and B memory cells. This doesn't show it, but another group just like this will be formed with just a clone of B cells that's ready to work now. So what's going to happen is we're going to get a clone of B cells just like this, a clone of memory cells, plasma cells are going to be formed. They're going to squirt out IgG, and memory cells are going to be formed. So this one's going to have an anamnestic response. So on second or subsequent exposure to green antigen, the immune system is going to just run into action with immediate maturation of these